Hello, everyone. Hope you're doing well today. So first off, I didn't intend for 9 and 10 to, EK's 9 and 10 to cause people sort of consternation about what do you draw, what do you make. These are really just looking at ideas related to genetics and sort of explaining why they are what they are. So in EK9, it says develop a model that illustrates each of the following claims about inheritance. When they say develop a model, it could just be like big A times little a, little a. That's a model, or coming up with a little Punnett square, stuff like that. Those are models that explain what's going on here. So when the first bullet says if a human male possesses a recessive allele for an X-linked trait and he reproduces with a female who has two dominant alleles for the trait, none of their sons will inherit the recessive allele. You can just set up a Punnett square that has like XY for the guy, and it shows that he has the recessive allele. So it could be like X little HY, and then on the outside of the Punnett square, you also have the mom. She's XX. She's homozygous dominant, so she'd be like big H, big H. And then you'd see when you fill out the Punnett square that any son would have a big H for mom, so he wouldn't have the condition. So that's all you got to do for that. The second bullet point says if two individuals who are carriers for cystic fibrosis have four kids, all four of the kids could be affected by CF. So CF is something that's autosomal recessive. So you could have people who are like big H, little h on the outside of the Punnett square, and you'd show that you'd have like 1 in 4 odds that a kid would get two little h's, and then you can multiply that out, a fourth times a fourth times a fourth times a fourth to show that all four kids would be affected. The third bullet, organisms that self-fertilize like many species of plant and like the hermaphroditic nematode, like a flatworm, C. elegans, produce offspring that are genetically distinct or not clones. If an organism is hermaphroditic, that means it has both male and female reproductive systems, so it can produce its own eggs and its own sperm. And egg and sperm will each undergo meiosis, and because they're each undergoing meiosis, you're going to get independent assortment, law of segregation, all that stuff from Mendel. So you could just, to develop a model, you could just explain that because each organism has independent male and female reproductive systems, meiosis is occurring independently in those structures. So you have independent assortment, you have crossing over happening, stuff like that. So you're going to get genetically unique offspring and random fertilization even. Let's see, last bullet. If the following statements are true, a gene has two alleles, one dominant over the other, there are two distinct phenotypes, one with the dominant, one with the recessive, then it follows that an individual of unknown genotype exhibits the dominant phenotype. That individual's genotype could be determined by crossing with an individual exhibiting the recessive genotype. Hey, it's a test cross, right? So if you have an individual that's got the dominant phenotype, you don't know whether that individual is homozygous dominant or heterozygous. But if you mate them with an individual with known genotype, a homozygous recessive individual, then you just look at the ratios to the outcome. If it's a 1-to-1 ratio of dominant to recessive in the offspring, well, then the parent with the dominant phenotype is heterozygous. So if you do the mating and all the offspring have the dominant phenotype, well, that parent with the dominant phenotype was homozygous dominant. So you can just sort of show that. All right, so that was EK9. EK10, what do we got? In real life, very few traits have inheritance patterns that can be predicted or explained by a simple Mendelian model. As we have discussed in class, the expression of genes are often affected by environmental factors. In addition to environmental factors, there could be other stuff going on. Okay, so the first item there are genes located on the X chromosome. These genes and the traits they encode are sometimes called sex-linked or X-linked. Again, males and females have different combinations or different complements of X and Y chromosomes, so they're going to be affected differently. If two genes are on the same chromosome, they're linked, so they tend to be inherited together rather than showing independent assortment, so that can alter your probabilities. Here's a big thing. Traits that are encoded by multiple genes are polygenic. So it's usually not just one gene influencing a trait, it's several genes. And those genes could be located on the same or different chromosomes. So that can really influence inheritance patterns. Some genes like blood type have more than two alleles and are codominant. If they're codominant, that means both proteins are being expressed. So you could have, you could be heterozygous. You could have two different versions of the protein and you make two separate proteins. That's, again, that's like having type A, B blood. And sometimes phenotypes result from incomplete dominance. So that means if you're heterozygous, you have one copy that's producing the protein and another copy that's not. So you make some of the protein. So you wind up with an in-between phenotype. All right, for this formative assessment, demonstrate why X-linked genes, traits showing codominance and traits showing incomplete dominance, can't be fully explained by Mendel's principles. So again, Mendel was law of segregation. You have two copies, but only pass on one at a time. And independent assortment, how one set of chromosomes doesn't influence how other sets of chromosomes line up. But the issue is, when he was looking at stuff, he ended up picking genes that behaved as if they were on separate chromosomes, only had one gene influencing that particular trait, and there were just two versions, like, purple flowers, white flowers, tall stems, short stems, that kind of business. So genes that are X-linked aren't going to show that same pattern of inheritance because, again, it'll affect males and females differently. Genes that are codominant don't match Mendelian patterns because both genes can be expressed at the same time rather than one being able to mask another. And then with incomplete dominance, again, you wind up with a third phenotypic option because some of the protein's being produced, so you wind up with an in-between phenotype. All right, that's the kind of stuff we were looking at for EKs 9 and 10. If you have any questions on any of that, let me know. Otherwise, hope you have a great day. Thank you.